Chapter 5 Molecular Genetics DNA Replication. The parental DNA makes one more copy of itself. This is called as replication. In mitotic cell division, the parental cell divides into two daughter cells. The genetic material DNA inside the parental cell is distributed to two new daughter cells. So there is a need for the doubling of genetic material DNA. One DNA is copied into another DNA by the process called as replication that takes place in the S phase of cell cycle. We have learned in 11th standard about cell cycle. In the S phase, that is synthetic phase, DNA makes a copy of itself. Replication takes place during the S phase of cell cycle. After replication, each DNA molecule gives rise to two DNA strands identical to each other as well as to the parental strand. The top one is parental strand. The two are daughter strands. The two daughter strands are identical to each other and both are identical to the parental strand. DNA is a macromolecule. It is made up of nucleotides. So DNA is nothing but polynucleotide chain. A nucleotide chain is formed by the addition of nucleotides like adenine, guanine, thymine, cytosine. When the nucleotides are added, a new strand is growing. This is a polymerization reaction. So this reaction is catalyzed by DNA polymerase enzymes. Now let us learn about the enzymes. Enzymes involved in DNA replication. Both prokaryotes and eukaryotes have different kinds of enzymes for replication. Prokaryotes have three types of DNA polymerases for replication. They are DNA polymerases 1, 2 and 3. The three enzymes cause the replication process within 38 minutes. Within 38 minutes, the DNA replication completes in prokaryotes like E. coli. The number of base pairs in E. coli DNA is 4.6 into 10 power 6 base pairs. This number of base pairs are copied, that is replication. This replication completes within 38 minutes. Okay. The DNA polymerase 3 is a main enzyme. The DNA polymerase involves in DNA replication. DNA polymerase 3 involves in DNA replication. And DNA polymerase 1 and 2, the first and second, are involved in DNA repair mechanism. DNA polymerase 1 is called as Kornberg enzyme. The DNA polymerase 1 and 3 the DNA polymerase 1 and 2 involve in DNA repair mechanism. What is repair mechanism? When the nucleotides adenine, guanine, thymine, cytosine are wrongly arranged, that results in complications. They are rearranged. The wrong arrangements are rearranged by the repair mechanism. In eukaryotes, 5 kinds of enzymes involved in the process of DNA replication. These 5 types of DNA polymerases catalyze the polymerization of nucleotides at the 3-OH of new strand. In the new strand, nucleotides are added at the 3-OH end within a short period of time. So this is old strand, left one, the right one is new strand. In the new strand, new nucleotides are added. Adenine, guanine, thymine, cytosine nucleotides are added at the 3-OH end. Error in replication. When there is error in replication, that results in mutation. What is mutation? Change in the structure of gene. When there is change in the structure of gene, that affects many activities of an organism. Replication takes place faster, at the same time accurately. Without repair, the replication takes place. Replication errors are corrected by repair enzymes such as nucleases. The replication is a process. Any biological process needs energy. The replication also needs energy. Replication energy is given by deoxynucleotide triphosphate. It acts as a substrate. It provides energy for the polymerization reaction, that is DNA replication. Let us see the mechanism of DNA replication in the next part.